Thank you very much for giving me the opportunity to present some data that I have from my research group. I have to say I am not part of the Digital Life Center, but when I read the announcement for this meeting, I got really excited because this gave me a really good opportunity to learn more, to meet people, but also I really hope also to create some collaborations. Because I come from the Department of Nutrition. I'm a nutritionist by training. I am trained in molecular nutrition, and I'm not a mathematics. So I really would like to collaborate with those of you that know mathematics modeling and how to reconstruct networks. So I will, the Systems Biology in Diet Intervention Studies is the title of my research group. And the vision for my research group is comprehensive understanding of fat quality and metabolic health. This is, of course, a vision. We are not come that far, but I really hope to come that far in the future. And because I have very short on time, I will not go very much into details, but I will hope to give you some glimpses into what we are doing and what kind of data that we have available. So as all of you probably know, nutrition is very important for our health. And it's been known for quite a long time that nutrients can directly regulate genes by acting as transcription factors or activating transcription factors that can regulate genes and gene expression. Many of these genes that nutrients are regulating, they are coding for enzymes, binding proteins, transporters, and proteins involved in uptake and absorption of nutrients. So of course also nutrients in that way also can influence the metabolites that are generated in the body. We also know that nutrients directly can regulate the epigenome by regulating the DNA methylation and also histone modifications. And of course our genetic background are also important for how we as humans can react on a specific nutrient. Since nutrients have all these important roles for our body, they can regulate a lot of biological processes which are important for our health. And we are mainly focusing on cardiovascular disease, but also we have looked into metabolic syndrome. And what we normally look at are risk markers, classical risk markers, such as triglycerides, total cholesterol, LDL cholesterol, insulin, and glucose, etc. And what we know when we are doing intervention studies that I am responsible for is that we see a large variation, individual variations in response to a diet intervention. So what I've mainly been focusing on is fish oil and fish intake and how that can influence CVD risk markers. I've also been involved in plant oils, particularly rich in omega-6 fatty acids, and also in different dairy products, and in krill oil, which is also another source for marine omega-3 fatty acids. And the aim or the goal of my research is to understand much more how these components in these food items can regulate genes and how they can regulate metabolites in our body, to really understand more how diet can influence and fat quality can influence biological processes and the phenotype. And the goal is, of course, today we give the dietary recommendations are basically given to the same advices to all people. But in the future, we believe that maybe we should move more into personal diet advices and give better, precise advices to the right time, to the right dose. To specific to the either individuals or to group of individuals, just because we know that people are responding very differently to interventions or to diet. So we are focusing primarily on because we are doing intervention studies, because of course there are a lot of other external stimuli that can regulate biological processes, not only nutrition. But by doing randomized controlled trials, we believe that we can, by the randomization, that we can take care of all the other external stimuli and just look at the intervention of interest. And what we are doing is that, and I will show you also some examples of from some two intervention studies that I have been involved in, we collect biological samples such as blood and urine, and we also collect the blood cells, which are the peripheral blood mononuclear cells consisting of lymphocytes and monocytes. And these we can do transcriptomics and analyze gene expression from. And we are doing metabolomics. We have mainly been doing in plasma. And of course, the goal for us, for the moment, we only have data on the transcriptomic level and the metabolomic level, but we don't have yet started to integrate these data. And that is really what is my interest and really focus for the future, if it's possible to integrate these data and in collaboration with mathematics people like you, that we can make we can create biological networks and so on, and really understand what's going on in the body after eating. So I will give you one example from a study that we performed. This was a double-blind, randomized controlled trial with fish oil, where we gave healthy subjects. These were in general, many of them were students, and we gave them, there were two groups that got fish oil. One group got what we call high-quality fish oil, one group got oxidized fish oil, and one group got a control oil, which is a high oleic sunflower oil. And the subjects were randomized, and then it was 17 giving this high quality fish oil, and then it was 19 people giving this high oleic sunflower oil, and also 18 giving this oxidized fish oil, because the primary outcome of this study was to really see if oxidized fish oil could have any negative influence on human health. And the outcome was triglyceride and oxidative stress markers, and I can tell you from this study, where we gave 1.5 gram EPA and DHA per day, we did not see any effects on neither of these groups on oxidative stress markers and triglycerides. But we continued to focus on the high quality fish oil and the control oil. And here we had an aim that we wanted to explore the molecular mechanisms behind the health beneficial effects of fish oil. And we isolated the peripheral blood mononuclear cells from the humans at baseline and after end of intervention, which was 7 weeks. And we also did lipidomics in a collaboration with Matteo Resik at the VTT in Finland at that time. And very briefly, I'll just show you the results from the use of Illumina platform. And we isolated these cells from using specific tubes, which gives very pure samples. And then we did limma analysis to analyze differentially expressed genes. And after quality control, normalization and filtration, 21,000 genes were expressed in these cells, and 470 genes were differentially expressed between the two groups. 236 upregulated and 235 downregulated. In addition, we did analysis to look at biological processes, and here we could identify among those genes that were upregulated, we could identify genes involved in cell cycle and DNA organization that were upregulated. We also did gene set enrichment analysis, where we could see that genes related to cell cycle, apoptosis, cell signaling, protein folding, ear stress response, and immune response was regulated. When we looked at the lipidome analysis, we were able to, and this was from plasma, we were able to identify 568 lipids, and 260 of these were identified. When we did the score plot analysis, we could see that the intervention groups were well separated by the lipidomic data after the intervention, and these were particularly the lipids that could be responsible for the separation between the groups, saying that the intervention were able to change these lipids. We did not see any change in total concentrations of lipid classes, and we did not, as I said, see any change on serum triglyceride, and we did not see changes in cholesterol, both total HDL and LDL cholesterol. But what we did see, what we found very interesting, was that there was an increase in the proportion of phospholipids and triglycerides containing the long-chain polyunsaturated fatty acids, which was not so strange because, of course, we gave them also marine oil rich with long-chain PUFA. So what did the results really mean? And then we went into cohort studies that has also used the lipidome to see if they could use lipidome to predict disease risk. And these are data that were published from the Framingham Heart Study, and where they used lipidomics on a subsample of the samples they have in their big cohort. And what they showed was that those subjects that had increase of these lipids, and how you read these lipids is that this is triglycerides, and these are the number of carbons, and these are the number of double bonds. So it means that when you have triglycerides contain 3 fatty acids, so of course these are long chain fatty acids with long chain with many double bonds. So all these were associated with reduced risk of to type 2 diabetes. And what was interesting was that particularly these triglycerides were also increased in our intervention with fish oil. So despite that we didn't see any change of triglycerides, but we did see a remodeling of the triglycerides, saying that in the future maybe it's more important to really measure the fatty acid composition of the triglycerides and not only measure the total triglycerides when it comes to disease risk, because maybe you can have the same triglycerides but you can have different disease risk based on which fatty acids that are actually part of the triglyceride. So that was the main finding from this study. So short, from this fish oil intervention study, we concluded that group of genes involved in cell cycle apoptosis and ER stress were modulated after intake of fish oil compared to the control group. And these processes and pathways are involved in normal cell function and activation of mononuclear cells for an optimal immune response, and may ultimately influence whole body health. We don't know yet what this really means, but at least we saw these genes regulated. And from the lipidomics data, we could say that the triglyceride signature generated by fish oil intake may reduce risk of cardiovascular disease and type 2 diabetes, because this has also been shown in another cohort that these lipids are also involved in or can also predict cardiovascular disease risk. Before I end, I also would say that we have another study where we have looked at where we have replaced saturated fatty acid with polyunsaturated fatty acids using a food exchange model to study the effect on cholesterol and inflammatory markers. And this was an 8-week intervention study, which we, in the X-diet group, where this X-diet group were eating food items rich in polyunsaturated fat, while the control group were continuing to eat the same food items that we were giving in the run-in period. And these were the same type of food, but it had a different fatty acid composition. In this study, we could show that there was a large and clinical relevant change in total cholesterol and LDL cholesterol. And what we are now very eager to look into is that we are now got the transcriptomics data that we are analyzing now for the moment, and we also have non-targeted metabolomics data that we are doing in collaboration with University of Copenhagen, and we also have targeted metabolomics with using NMR technology. That is also data that we have now got, which we have got from another collaborator. So these are the data. We still don't have the results, but we are very eager to see what they show. And so the whole question is, can we integrate this data set and understand better the effect of fat on cardiometabolic health? And this is really something I hope I can get some collaboration on with you. So then I just stop by thanking the people that have been involved in this project, and yeah, I'm open for questions.
Thank you very much for this interesting talk. So we have time for one question. Yes?
Yes, very interesting study. I was wondering if you have analyzed the composition of different lipoproteins,
Yes.
and you see the fatty acid change on each lipoprotein. Is it on polymicron or LDL or mostly HDL?
Actually, we have on this, in both these studies, we also have lipid subclasses. So we have the size of the different lipoproteins. We have the composition of lipids, and we can also see that that is changing. But I don't recall all the data, but we have those data as well. Yes.
Yeah, thanks. So it's So they are mostly not changing.
Yes, they are changing.
They are changing.
Yes. Mm-hmm.
Yeah, thanks.
Yeah, let's thanks the speaker again.